Thank you, Helga. Good morning, everyone, and welcome to my presentation, which is titled Optimizing Fruit Storage, and particularly on the convection, diffusion, and adsorption of 1 MCP gas in a cool store, which nicely follow the previous presentation. Actually, this work has been done a long time ago while I was in Belgium as a PhD student. And we found it's a nice complement to the symposium, and it may provoke some discussion. And here I will start presenting to you. I would like to start with this slide because it nicely shows the drama between ethylene and 1 MCP. It's on tomato. A tomato is a nice fruit to show how these two gases have an effect on the quality of the fruit in terms of firmness. You can see how this 1 MCP technology is very important for the industry. I don't want to stay more on this slide. We also saw listen to the previous presenter on how 1 MCP is important on other physiological defects as well. What I would like you to have a look is this map, to where the 1 MCP that we put in cold store will arrive. And so you know how 1 MCP is applied. We put a powder in say a bucket of water, and the 1 MCP from the powder is released to the air, and the 1 MCP in the air will diffuse to the fruit or any solid material to that matter, and it diffuses to the void space in the fruit, and it may bind to the ethylene binding site, which we say okay, that is what we need, or it may bind to some other binding site, which are non-specific, non-target binding site, which in a sense is a loss, or some of the 1 MCP may simply reside in the free void space, and in the subsequent ventilation case, it will come out and release to the atmosphere. So I would like you to remember this map, and then I will show you how we use the modeling technique to make an inventory of the 1 MCP that we put in the cool store. So the main question then was, are we treating all the apples, say, all the fruit in the cool store everywhere equally? That means, is the distribution of this gas in the cool store is uniform? And as I already told you, there are non-target binding site in the fruit, not only those, but there are non-target solid material in the cool store as well, be it a wooden bin, a plastic bin, or cartons that we use to pack the fruit. So those material may participate in the 1 MCP uptake, and how this would affect the treatment. That was the other question to answer. And is a room size and room shape has an effect on the distribution of this gas. And sometimes in the storage, we put plastic liners to prevent moisture loss from the fruit. Will that affect the gas distribution? Is also another question. And the dose aspect. Dose aspect is quite tricky. We should stick to a prescription, but we try to quantify how much 1 MCP we really need to satisfy the ethylene binding site. Technique that we use is experimental technique, and in stage-wise experimental technique, we start with jar tests with a precise measurement, and then small container level to a pilot cool store level and commercial cool store level. And each of these stages were followed with a mathematical model to mimic the process, the physics of, or the mechanism of the 1 MCP transport. And then we tweak the model to perform what-if scenario and to measure how much 1-MCP we need to satisfy the ethylene binding site, how much left over in the air, inside the pore space, and gone to other non-target solid material. So the model starts from simple material level, goes to fruit level models, package level, and cool store modeling technique. We use commercial software, industry standard, so that the result that we get is credible and more accurate. So I don't want to stay more here, and these are the workhorse, the main mathematical equation that we use to define how the gas transport in a room filled with apples. What you see here is a equation that will tell us how 1-MCP travel in space, in air, and in the solid, be it the fruit or the some material, solid material that we see here, is captured by this equation. And the whole modeling is to solve this equation for a given dose. The experimental setup that we use at this jar stage look like this one. We create a 1-MCP with a known dose in a jar, then we transfer it to a jar that contains the sample material. Sample can be any material that you can be interested. For us, a plastic piece, a wooden piece, and fruits, and also we put fruit and those material together, and then we measure how 1-MCP is depleted by the fruit alone and with combination of those material. And you can see here the 1-MCP concentration in the head space. And this equation is, this model is placed in those equations that I showed you, and then those parameters are solved, and we got this material properties which describe how the different material react with the 1-MCP gas. As you see here, we have the two bean material, wooden bean material, oak wood and poplar wood. They indeed have a huge 1-MCP uptake capacity, with the poplar wood becoming more porous. It has a lot of 1-MCP uptake capacity. A plastic bean, though they have some capacity, but the diffusivity to this material is very low, and we can here say probably plastic have very little impact on the distribution. Card lining is in terms of 1-MCP uptake, is more than any other material when you compare it with the apple fruits and with the other material as well. Especially what we do here is we measure the uptake of this material as dry and wet. A wet material take more 1-MCP than dry material. And the fruit, we saw Golden Delicious and Jonah Gold, but we don't see any difference, any statistically significant difference between them. But what this result tell us is that those material that we say a non-target material, the wooden bean and the cardboard, are indeed competing for the active ingredient in the cool store. So next, the tracking of the 1-MCP in real storage, equipping us with those material properties and those equations. We do this experiment, which I show you here the setup, and the corresponding safety model is shown here. These are sampling technique that we use. A pipeline goes to the system, and at the different spot that you see here, we take gas samples, and those gas samples are measured with GC for 1-MCP. And that sampling and measuring is done an interval of time. We have the model and also the experiment as you see here in terms of the air circulation, how air circulation affects the distribution and how different material affect the distribution is measured here. As you see here, a plastic bin with plastic lining and wooden bin. Though plastic lining have no effect on the 1 MCP depletion, as you see here, wooden bin has a huge impact. And so box material we see has a huge impact on the distributions and airflow and other factors. The next stage is to scaling up this analysis to a large commercial cool store. And the problem here is it's almost impossible then, I think now as well, to model how 1 MCP will enter to individual fruit, to individual packs. So we need to use mathematical simplification. And for that, we use what is called a porous medium modeling technique. It's simply to simplify the application of the techniques that we started. And this technique helps us to simply consider a box filled with fruit as a box. But the property of this box is dictated by a measurement on a single box, and as you see in a bin, of how the air flows through it and how a pressure drop look like through it. So that will help us to describe each bin in a cool store as a porous domain, and we'll be able to model how 1 MCP distribute in the big cool store. As you see here, this is a 1 MCP generator, and the cooling fan is here. As you see here, the generated gas follows the airflow stream, and at early time, some of the bin near to the generator may show a little bit higher concentration, but don't worry, this is at early stage, and when time goes, I will show you everything goes uniform in the cool store. These show how room shape and room size will have an effect on the distribution of this gas, and sometimes due to the use of forklift and some of the pallets here is missing, will that have an effect on the distribution? Can be also be assessed using this technique. And what we saw here, be it room size, shape, has minor effect. We go down to a very small difference between region, and what we see here is an exaggerated difference of the gas distribution. But normally it can be said that the normal airflow levels that we have in a cool store may be sufficient to take care of that non-uniformity. In terms of uptake as well in the large cool store, we'll be able to compare how wooden bin and plastic bin will function. Broken curve that you see here is for wooden bin, a full curve is for plastic bin, and at different 1 MCP dose, we be able to trace where that 1 MCP goes, as I showed you initially. Does it go to an ethylene binding site, or in the pore space, or free in the air, in the wood, and so on and so forth. As you see here from a previous work by Blankenship and Sisler, I think, we know the total amount of 1 MCP binding site in an apple or in a fruit is about this value. And as you can see here, all treatment, say 0.3 ppm, will have much higher than required for 1 MCP binding site. If it is satisfying 1 MCP binding site, total amount of 1 MCP, ethylene binding site, a total amount of 1 MCP that we put in a cool store is much, much greater. So here we have a summary of a distribution of the gas for a plastic bin and a wooden bin. As you can see it here, a wooden bin takes something like 31% of the active ingredient from a treatment. So the fruit got the required amount, but we lost a lot amount of 1 MCP to the wooden bin. And also we use our model to see how room filling will affect the dose. Technology is invariably prescribing 1 ppm or 0.5 ppm for a cool store, but we saw from our models that highly filled store and partially filled store may have different dose, but that should be prescribed or given acknowledgement from the industry. But our model showed that for partially filled room, it's possible to have lower dose. So as a conclusion, what we saw is the wooden bins and other plant-based material have a huge impact on the uptake of 1 MCP, and the industry can take note of this effect of such material. And in terms of the simple calculation, we saw that the ethylene binding site, the site that we are interested to occupy and prevent ripening and the senescence, are far less than the amount of 1 MCP that we are using. We can estimate that is 2,300-fold higher 1 MCP that we put to the system. So this may raise a question to a developer of such chemical: is it possible to be more selective so that we address those ethylene binding site than simply treating the whole thing? What we saw here as well, a previous work show 1 MCP up to 0.02, they do up to 0.02 ppm. They only miss significant or measurable effect at 0.02 ppm, but our model show even 0.02 ppm is higher. What we notice is probably the 1 MCP, the ethylene binding sites are in endoplasmic reticulum inside the cell, and the 1 MCP that we put should pass through the cell wall, and cell wall will take more 1 MCP, and we end up with no treatment at lower dose. So the amount that I show here should not be followed. We should strictly follow the industry standard, but this just to invoke a discussion, and I simply call it, are we treating more than possible for non-target material as well binding sites. So as a future outlook, I would like to implement this model, increase its capability to see it with DCA and see it with CO2 and O2 distribution as well. And I would like to thank the following industries for their support, and would like to thank my supervisor, Professor Bart Nicolai, and Peter over here. Thank you.